So, page 1041 talks about the external genitalia of the female versus the male. What's the most obvious difference? Other than that, huh?
I can't help.
See, I'm glad I'm not the only one. The female's reproductive organs are more internal, so there's an actual separate entrance to the female's reproductive system, and that's what we see externally. So there's that additional structure externally. Also, the urethra is very small because there's no need to share that passage with the reproductive system. Next thing they talk about in the textbook is mammary glands. Mammary, what are those doing for us? Milk. So milk production, because female is responsible for not only gestation of a new individual, but for nourishing that new individual as well. So female mammals tend to have some sort of apparatus to produce milk to feed the young when they are born, and that's what breasts are for. The mammary glands are actually what kind of glands? Yeah, the sudoriferous glands. Remember when you studied skin way back when? And we had a mention of a modified sudoriferous gland, and we saw two things, ceruminous glands and mammary glands. So they are going to produce milk, and that's what those are for. Unfortunately, because of the highly vascular, lots of adipose tissue, lots of connective tissue regions in the mammary glands, there tends to be a higher risk for abnormalities with cellular formation and breast cancer. So there's a little discussion in your textbook on breast cancer and detection and treatment. So as we get older especially, we see higher incidence of formation of cancer in the breast tissue. One of the tests you can have very quickly to see whether there's changes in density in breast tissue are mammography. And when do they typically suggest you start having mammography on a regular basis? 40. Exactly. If you have a history in your family and there tends to be a high incidence among females in your family, then they suggest it earlier. So, but one of the problems is it's not necessarily going to happen after 40. And there's a very high incidence of breast cancer showing up in females that are a lot younger, especially nowadays. So it's important to do regular breast exams, and if there's any changes, to see your doctor. So they talk about diagnosis, treatment with radiation therapy, chemotherapy, surgeries to remove, and because there's a lot of lymphatic vessels in the area, that's another issue with malignancies and breast cancer. So if you see some changes or feel some changes, go to the doctor. We talked about spermatogenesis when we talked about the production of what? Sperm. And what was my abbreviated version of spermatogenesis like? We have one immature cell that's going to do what? Go through meiosis 1, go through meiosis 2, and what are we going to end up with at the end? We end up with 4 gametes, 4 sperm in the male's reproductive system. When we look at the female's reproductive system, it's a different story. The process of oogenesis is going to be a little bit different. We're still going to undergo those same splits, but the end result is going to be different than that of the end result of spermatogenesis. For every immature stem cell, and that's called an oogonium in the female ovary, we are going to end up with how many mature oocytes or ovum? 1. What happens to the other? Huh? The oogonium at the top is the immature cell, that stem cell. The ovum, or the oocyte, is that mature gamete that's produced from that. So we're going to go through the same types of splits, but we're going to see a very different cell at the end. The nice thing about this diagram is it shows you how different that cell looks on the side. When you go to lab and you look at your histology for the reproductive system, be sure to look for these large follicles that start to form during a female cycle. And during the female cycle production of this cell, we're going to talk about different stages of maturation of these follicles and what we're going to see inside there. So, in the beginning, we have that primordial follicle that sits and waits for hormones to tell it it's time for the cell to mature. In infancy and adulthood, or childhood, we start to see that primordial follicle and primary follicle come to appear inside the ovaries. So they mature a little bit during puberty, when female hormones start to reach certain levels. And we're at the point in puberty in the female for these cells to mature each month. So we start down at the primary follicle, which for every monthly cycle, we're going to mature into a secondary, and then vesicular, and then eventually that follicle that's left behind with the cell that's been produced by meiosis, again, two-stage split, is something called an oocyte, which is going to be released during a process called ovulation. The cells that are left behind in those follicles have a job to do. And they are actually going to produce hormones that are going to help maintain that endometrial layer that we mentioned last class for a certain period of time to allow, if it happens, the implantation of what? Fertilized oocyte, fertilized ovum. Okay? So at that point, and we'll talk about what it's going to look like at that point, that group of cells in the corpus luteum is going to help to produce hormones to maintain that layer so that if implantation takes place, we can start the process of gestation.
Meiosis tube will not happen without sperm, right?
No, it will happen.
It says in the book that it will not happen.
No, no. Well, you have that first stage, prophase 1, prophase 2, and then metaphase 2 it happens in the secondary oocyte, so we have that second.
No, it says that meiosis tube completes only if sperm penetrates them.
It completes, but we're going to start it up.
Oh, that will start, but it will not
It's going to stop where?
in
Metaphase. So the second metaphase is where we're going to hold until fertilization takes place. Metaphase is lining up. So we have the copies of genetic material that line up and are ready for the split. So we're going to arrest there.
But why they are arresting this? I mean,
That's the way it happens. I don't have an answer to that one.
I know.
But that's where it stops. And then once sperm has entered, we'll finish up with meiosis 2 and form with that extra genetic material something called a polar body. That polar body is something that we just reabsorbed. Okay? So this is what it looks like under the microscope. Again, pay attention when you get into lab and try to find some of these primordial follicles versus that mature Graafian follicle. We see that guy down at the end. You can see the cell surrounded by a group of cells for protection called the zona pellucida, and that's what the sperm has to penetrate before the genetic material is injected into the ovum. So when we talk about that in a moment, we'll see. So during any maturation cycle, and that's where my exciting diagram that I showed you before is going to come into play, we're going to see several different hormones that are going to establish that cycle, the maturation of gametes from the immature primordial follicles. So in your textbook on page 1047, we begin the discussion of hormonal regulation of the ovarian cycle. So during that time, what are we trying to do within the ovary? During the ovarian cycle, what are we trying to do? Or trying to mature. So we're stimulating with a group of hormonal signals, the maturation of that eventual ovum from that immature cell. How do we do that in males? Yeah. Well, how did we cause the cells to start to mature from stem cells in the male's testes? Some of these same hormones, right? Gonadotropic releasing hormone. Is that here in your diagram? Follicle stimulating hormone. Is that here in your diagram? Luteinizing hormone. Is that here in this diagram? So it's very similar with respect to the hormones secreted by the central brain regions that are going to stimulate the maturation not only in the male but in the female's gamete production. So we see the same hormones involved in the maturation of that immature oocyte. Very similar, not exactly the same. So gonadotropic releasing hormone from where? The anterior pituitary gland is then going to get into circulatory system and going to stimulate what? Release of who? Follicle stimulating hormone, luteinizing hormone. We're going to see different feedback systems involved. That luteinizing hormone surge is going to trigger eventually ovulation, the release of that now much more mature oocyte or ovum. And if fertilization takes place, that can then implant in the uterus. So just a basic understanding of the hormones, which again are very similar in the male's maturation process and in the female's maturation process. What I want you to pay attention to is the result of these hormones. Because not only do we have the similar hormones up at the top of this diagram, luteinizing hormone and follicle stimulating hormone, but we also have two other hormones that are going to help with the process in females as well, and that's estrogen and progesterone. So the next thing they talk about is the uterine or menstrual cycle. Again, this diagram is going to tie everything together and hopefully make you understand what's going on during that 28-day cycle. So during that 28-day cycle, we start it with day one. And day one is basically the end of the cycle, beginning of a new cycle. Day one is getting rid of the old layer, which we're going to spend another 28 days working up to, because what has not happened? Yeah, no implantation has taken place. So day one starts with
People who even had fertilization, but still had a menstrual phase.
yeah, that happens too. It's rare.
Why?
Just the way their body reacts.
But if they will rid of endometrium,
No, sometimes it happens, the implantation takes place, they're able to establish enough of a chorion and the base stem of the placenta so that when the endometrial layer goes, that has already been established. So it's very rare that it happens, but it does happen.
with my mother-in-law, it happened all of her
Yeah,
before pregnancy.
and again, it's not the common, it's rare, but it does happen. And, normal pregnancies can happen that way as well, which I think is kind of a ripoff because that's one of the only pluses of pregnancy. It's not having to deal with, you know what I'm saying? So, yeah, it's a ripoff when that happens. But it can happen. It's rare. Yeah, I know.
She was pregnant before she started to feel moving.
Right.
When she was 5 months.
Yep. So it does happen. It's very rare, but it does happen. So, menstrual phase, that's the beginning, day 1. When we shed that old layer and we're working towards creating another mature cell to be ovulated and another brand new endometrial layer. With the help of estrogen, we're going to help that layer start to form again. When we see a spike in luteinizing hormone and follicle-stimulating hormone, that's the end of the proliferative phase. I hate that word. You know, in my research job before I came here, you know what I used to do? Proliferation assays. Yeah, and I had to present my data to my group all the time, and I can't say that word. So anyways, at the end of the proliferative stage, we see a spike in follicle-stimulating and luteinizing hormone. That's when we see what happened? Ovulation. So the release of that almost finished meiosis to oocyte, ovum is another name. We continue now with the help of progesterone to continue to do what during the secretory phase?
To keep this layer ready for implantation if it will happen.
Exactly. So we're going to continue to build the endometrial layer. We're going to add to it a whole bunch of endometrial glands, which are going to aid in the formation of chorion and eventually the placenta if implantation takes place. So implantation will take place somewhere around the center of the secretory phase. If it doesn't take place, we start to see what happening. What happens to the hormones? Yeah, and that's why I like this diagram, because you can actually visually see it. We also, if we follow up above that diagram, we see the corpus luteum, the cells that are responsible for making these hormones, right? What happens to it? It starts to degenerate, and that's why we start to see the levels of estrogen and progesterone going down.
You know the post-menstrual syndrome?
I was just gonna say.
Okay.
So, if you look at these hormone spikes, they're quite high, and some women are very sensitive to this giant spike in hormone levels in their bodies. What's that called? PMS. What's PMS? PMS! What's it stand for? Premenstrual syndrome. You see why they call it pre? It's before day 1, yes? So we see that spike around day 14. We see another spike around day what? 20, 25, whole bunch of estrogen, whole bunch of progesterone, LH, FSH, hormones all over the place, yes? And again, sometimes females are very, very adversely affected by such a jump in their hormone levels, and that's what PMS is. It's not fake. Some people are very, very debilitated by that spike in hormones.
Unfortunately, there's no cure. I cry.
You cry? Yeah.
That's a cure.
I'm Italian. I cry all the time. But yeah, your emotions, your yeah, or your temper. Some people lose their temper really quickly. You get what?
I tweak out in class.
You tweak out.
Teddy bear!
Not a good day for teddy bears during day 20. So we see a big spike in these hormones. Again, these are hormones that are helping to mature those oocytes for release and then maturing the endometrial layer. So understanding what's going on in this diagram and how it all ties together gives us a good perspective of the ovarian cycle, what's going on during that 28 days. The effects of different hormones like luteinizing hormone, follicle stimulating hormone, gonadotropic releasing hormone, and then the effects of estrogen and progesterone. Those are things you should understand for the exam.
Create, tablets, drugs are helping those hormones to be a little, especially because you're not going to be pregnant if you're, trying to. Why they don't? They
That's what you could do, Mila. You could create those drugs and you could get rich because all the women in the world would love you. So.
Like birth control, I know you have that, you still have the period phase, but doesn't that, like, because some people, like doctors will recommend continuous cycling where you don't have your period, doesn't that keep your hormones at, like, level? Like,
Yes.
the whole
And what
time?
does birth control prevent you from doing?
Ovulating?
Yeah. Because it keeps certain levels, estrogens, progesterones, up higher, so it fakes your body into thinking it's in day 20. So it'll never go into day 14. So that's what it does. It fakes your hormones out.
It takes your body into thinking you're already pregnant.
Correct.
Oh.
So it fakes your body hormonally into thinking you have had implantation in between day 20 and day 25. When you take the break, you're already past day 1 through day 14. You take your break, you do what? You shed your layer, you start at day 1. You only give it enough time to start the shed, then you fake your hormones in again, and you never get a chance to start doing what? Yeah, they start that maturation process in the follicles of the ovaries.
What's happened in that time with follicles? They not ever mature. They
No.
were ever. Would they affect the whole process in the ovaries? Because
The
I know
process-
Some people using those,
Birth
drugs,
control?
Yeah, birth control. They afterwards they will not get pregnant because.
There's such a touchy, yeah, but it might not necessarily have been that. It might have been the fact that they wouldn't have been able to get pregnant in the first place. They weren't present. So, yeah, that's a controversial.
The process which naturally happens.
Well, it masks the process. Your question is it going to affect the process once you're done? Who knows? No, I can't answer that question.
I got off the pill when I was 35 and had the baby at 40. Now, are you eliminating, because you said that when an ovary is released, you build up scar tissue. If you're on birth control, you're releasing the ovaries, so you're preventing the scar tissue?
Yep.
Now,
And you might be prolonging your what?
ability to get pregnant?
Yeah. Or you might be prolonging or delaying menopause
Right.
because you have less scar tissue on the ovaries.
And so, like, you said that, like, a woman has, like, obviously you're not going to use all of the eggs that
Correct.
you have, but you're not actually releasing them, so they're still just hanging out in there. So you're not wasting them.
No. And if there was a way, if
They don't get any
What do you say?
They're not getting
right.
younger, though.
They're not getting any younger.
But and the
Those
thing
haven't
is,
matured yet.
is there a way to maybe take those and use them
Later.
later?
Yeah.
Okay, but how do we mature them outside the body?
There's a lot of side effects of those.
Yeah, exactly, and that's the issue. If we could invent a way where we could take, say I'm done with my reproductive, I have had all my children, wouldn't it be nice if we could then take your ovary or one of your ovaries, bring them into the lab and mature those follicles outside your body?
Why don't you
We
go?
can't do that
Why?
because we haven't been able to do that scientifically.
I'm going to donate.
Well, to do that outside the body. So again, boy, if you could do research, yeah, donate your ovaries to science and see. But yeah, we haven't developed that technology yet. We still need the body to do all of that.
Somehow come up with a way to have those hormones, like, enter the ovary that's outside of the body.
Yeah, exactly. To affect it in
Somehow
the
figure out
same
a
way.
to extract those hormones.
Well, it's not that easy because it's, you know, there's such a fine balance. You remember, you're having delivery of these hormones at certain levels from the circulatory system. It's not like, it's very, very difficult outside the organism. You can take an embryo outside the organism, but the female has to mature those oocytes. Those can then be taken out, fertilized, and frozen and saved for later. But we still haven't developed technology which would allow us to do that outside the body.
Like when a woman's eggs are, because I know that they do have like fertilization, like where they, like you said, they fertilize it and then they implant the fertilized egg already.
That's in vitro
So
fertilization.
the egg is already matured when they extract it.
Well, it's in that,
Anymore. It's a zygote. It's already baby there.
right, because they take that mature ovum.
But how do they get that? They take it from the tube.
No, they take it from your ovaries.
And then
They have to go in laparoscopic surgery, and what they do is they pump you up with so many hormones that you actually have
use more than
a lot more primary follicles produced than you normally would,
and it's not
and they harvest those,
it's not a guarantee.
and then they can fertilize them outside the body, and then they can take that blastula, which we're going to talk about in the next chapter, and that's what they
So
implant.
they pump you up with the hormones.
Correct. To mature that oocyte.
Okay.
We can't do that outside the body.
Right.
That's what. Does that make sense?
Yeah, I was just a little confused because I know you said you can't do the maturation process outside of the body, but I know that.
You can. The fertilization can happen, yes. And then that ball of cells, the blastula, which again we're going to talk about next, that can then be implanted.
And usually they do like 3 or 4 at a time.
Correct.
That's like multiple.
Because chances are, and again, the natural process of that cells tumbling down the uterine tube is very important, and sometimes they can't replicate that artificially. Yes?
I have a question. I just want to go back just for a question. When you talked about follicular phase of ovarian cycle,
Mhm.
what exactly do we need to know from that page or that
What page are we on?
It's ovarian cycle, 1045, page 1045.
10:45.
Ovarian cycle.
Oh, yeah. The hormones.
What do we need to know?
Look at, remember that figure, where the heck is the figure?
This picture or what page?
No, it's on page 27, figure 27.21 on page 1048. So that goes through some of the different hormones being
It's
produced.
not hormones, it's about, this ovarian cycle talks about like how
Yeah, that's on,
motile follicle becoming primary follicle, then secondary follicle.
right.
Do we need to know the all details about how?
Well, you just, again, that general information, that was also in the events of oogenesis. But if you look at page 1046, and you see figure 27.20, that's where you see all those terms coming in. But yes, you should know primary follicle becomes
Just how it becomes
secondary,
details
exactly, secondary follicle, late secondary follicle, everything from that diagram we discussed, okay? Know that we're kind of halted in meiosis 2 before fertilization takes place, and that the end result of oogenesis is one ovum and what?
polar.
Yeah, those little polar bodies. Again, those are kind of reabsorbed by the body and kind of useless cells that are produced.
There is genes information in this.
Correct, but they're not functional. They're not functional, and they won't last very long, and they degenerate. You can't fertilize a polar. You can't fertilize a polar body, because basically what's in the polar body is very similar to what's in the sperm, just the genetics. You need all the other parts to be a cell, right? And that's what we're going to find in the ovum, the mitochondria and all that stuff. When we talk about heredity, we'll see mitochondria has its own genes, its own DNA. And where does your mitochondrial DNA come from? It comes from your mother, because that's where the mitochondria was in the ovum before fertilization took place.
So our mitochondria in our cells are genes from our mother.
Only your mother, not your father. Because what? Well, we had more work to do, as usual. As usual. Okay, so the other thing I do want you to know from this chapter is a basic definition of the sexually transmitted infections that are listed on page 1053 and 1054. Many of them are some sort of bacterial nasty, and some of them are viral nasties. So just a general definition of those guys. We do have a little parasitic nasty over when we talk about trichomonas.
This is parasites?
Mm-hmm.
Really? Like worms?
Your parasitic, yep.
Like, wow.
Okay? So these sexually transmitted, you can't get them from a toilet seat. You cannot get them from a swimming pool. They have to be sexually transmitted through the sexual process. Yes? They discuss. Say it again, hon. We're going to talk about that in the next chapter. Next chapters.
Maria, what's about genital warts? What is it?
Genital warts is caused by herpes, and that's exactly what it is. Warts in the genital region. Oh, HPV, I'm sorry. You can also get genital warts from herpes, too. But, yeah, there's another viral nasty besides herpes called human papillomavirus. There's also a high incidence with people who are infected with human papillomavirus of cervical cancer. So one of the things that's new on board with respect to prevention for women is vaccines for HPV. Yep.
You want them to do that in their teenage years now.
Yep.
Now, men can carry HPV, but
Correct.
they don't usually show symptoms of it.
Correct. Yeah, and men, and because they're sexually transmitted diseases, men and women are affected by these in different ways.
I just, when I read it, home, I was like, how come people would not have any signs of diseases? Who says 70% of cases, women or men would not know that they have that.
Because, well, it's when we think of disease, we think of enough organism in your body that will then create adverse effects.
So you can't see it until you have an outbreak.
Right.
You won't even know that you have it until you have an outbreak. Because my friend actually got it from somebody who she had sex with, and she didn't know that he had it because he didn't tell her.
Mm-hmm.
And so when it was the first time ever being sexual, ever, and she got this genital herpes from this guy because he didn't tell her she had unprotected sex.
Yep.
So now she has it for the rest of her life.
Correct. Correct. And they may not know they have it until what?
They have an outbreak.
Or they have enough organism in their body to cause mal effect. So if you have a virus, for example, sometimes you can pass that virus to many, many people before you even start to feel the ill effects of the virus, because there's not enough in your system to make you sick yet, but you're still contagious. Oh yeah, you're infectious. But chicken pox, perfect example. 7 to 10 days. So you don't know until you start what? Getting the chicken pox that you have it. But you may have, exactly, you may have infected quite a few people. Right. And that's why the vaccine is such a great thing, because it can help.
Are they now recommending the vaccine for males as well?
I think so. They started after the first development, was kind of rough, but yeah. All right, my friends, so that's the end of chapter 27. Chapter 28. Now we talk about pregnancy and human development. So after fertilization has taken place from egg to zygote, we now are going to take that one cell that is basically a combination of two half cells, two cells with 23 chromosomes worth of information, bring them together during the fertilization process, and create a brand new individual with 46 chromosomes. So you come from how many cells? One. Because them on their own, exactly, them on their own, they're really not going to do us any good until they come together to create that brand new individual. So fertilization in this diagram gives us the concept of what happens size-wise to that one cell within about 12 weeks. So that one cell is going to then, in 12 weeks, become a fetus. Different books, different time frames, 30 days-ish, 2 months-ish. It depends on the textbook, but they call this ball of cells a different thing at different times. So when you first have fertilization take place, you are called a what? A zygote. So a zygote, the first cell of a new individual, is the terminology we use. Now, sperm must enter the female's reproductive system and travel up through the fallopian tubes in order for fertilization to take place. Once they get there, they have to penetrate that barrier of cells, the zona pellucida that we saw earlier in the previous chapter. And they do that with what? Correct. The little sac of enzymes in the head of the sperm called a what? A flagella is going to make me move. Acrosome. So acrosomal reaction and sperm penetration on page 1066 explains how that enzyme is going to then break through the zona pellucida and eventually allow the genetic material inside to enter. Sir.
Serious question. Does anybody remember 3 Men and a Baby?
Yes.
Does the lightning happen?
That was a good movie.
Remember that?
Yeah.
What about the beginning of Look Who's Talking?
Yeah.
It shows the sperm way down and like right.
Fertilizing the egg. You know, it's like computer graphics, I guess. And then once the sperm wiggles in, there's like lightning. Ripples across the egg, very dramatic.
Well, it's a good representation because what happens when one of these acrosomes starts to wear away the zona pellucida? We do have a block that occurs across the plasma membrane of the ovum that stops other sperm from doing that. So that we prevent what? More than one sperm from entering. It's not going to be twins. If more than one sperm enters, we're going to have how many chromosomes? Yeah. So we're going to have yeah, that's not going to work. Yes?
Would it be a miscarriage or would you have
It just the cell wouldn't start to split. It might do something. It might even get one split in, but it's just going to end it there.
it would end up becoming a child?
Not a human child, no. Nope. Nope.
And this is kind of breaking stuff.
Yes, exactly. So that reaction, and again, I'm not going to make you know the details of it because there's just way too much material right now for you to shove in all your brains. But once one sperm, one acrosome has started that reaction, that acrosomal reaction, we're going to block the rest from penetration. And that's what they discuss under the block to polyspermy, too many sperm from entering. And we see that in this diagram here. That penetration is going to do what? Yes, exactly. It's going to cause the cell to then complete meiosis too. So we get stuck during what? Metaphase hanging out in metaphase 2, waiting, and then once penetration takes place, we continue the process. So what we see are what we call pronuclei. Sets of genetic material. One is from the male and the sperm that was deposited in, and one is from the female. That extra group of genetic material is ejected from the ovum. And what is it called? That's another polar body. So that extra set of genetic material that got stuck during metaphase 2 is ejected from the cell as a polar body. So there's actually 4 polar bodies that get ejected from the cell
So two
maturation.
from one, right? Or one from
We get one from
one.
one, and we get how many polar bodies in the end? 4. So 3 polar bodies before fertilization.
Body one will split as well.
Correct. And then we form a 4th polar body after fertilization has taken place. So that extra genetic material that didn't get pulled away will get pulled away in the form of a polar body.
How is it possible, because I heard something like that, is it possible for women to get pregnant from 2 men at the same time?
Yes.
How?
2 different, but they have to have 2 different oocytes have to be present.
Oh, 2 different?
Yes, not 2 sperm cannot enter the ovum and then have a normal zygote produced. So,
That would
yep,
be twins then?
it would be what twins?
Not identical.
Not identical, but fraternal.
Fraternal.
So yes, has that happened? Yes, probably more than we know. Huh?
It happens from too much sex.
Too much sex. Well, yeah. Okay, so we see in these diagrams the progression of the cells as they go through their division. Those pronuclei will eventually combine. No, just the general information. And again, we see this diagram on page 1070, which shows us the progression of the genetic material as it goes through that first division. So sperm will enter, then sperm and ovum nuclei are going to swell. Then we see those swells, which we call now pronuclei, are then basically going to come together to do what to that genetic material from the both nuclei? Combine them. So then we are going to have a whole brand new what? We're going to have a whole brand new cell with a whole brand new beginning, which we will start to then take those 2 pairs of chromosomes, just like we have in the mature cell, 46 chromosomes, and that will then start to divide. We'll get rid of the excess genetic material in the form of polar body.
Maria,
Yes, ma'am.
How when those chromosomes will, get together,
Mm-hmm.
and replication will take place, right?
Correct.
Replication. So, with the regular replication, that cell would divide, right?
Correct. So it's just like regular replication now.
Yeah.
Once the 2 sets of chromosomes come together to form a cell with 46 chromosomes, that's the first cell, zygote. Then what's going to happen? Mitosis, like crazy. So that one cell will start to undergo the process of mitosis at a very rapid rate. So if you look at this diagram, we see that after fertilization has taken place, and just one little thing I want to point out, when ovulation takes place in a woman, the ovary and the fallopian tubes are not attached. So how does the ovum get into the fallopian tubes? So what we have is an ovary like this and fallopian tubes with these little extensions on them.
Are they like cilia?
Not cilia, but big, more like villi, okay? So, or fingers. They're called fimbria. And what's going to happen is this motion is going to take place. Exactly. So the ovary, sorry, That's all I got for visuals. The ovary is going to eject the ovum, and that wave-like motion of the fibria is going to attract it into the fallopian tubes. Does it miss? Sometimes? Yeah.
Is that why they say that sometimes you can feel like your sometimes feel your
Some women, if they have cysts on their ovaries or some other disruption going on with their ovaries and fimbria, they can actually feel when they're ovulating. So they'll get pain when they ovulate.
somebody said I think the doctor you can sometimes feel like almost like a little tugging, like very slightly.
No, and there's ligaments and tendons that hold all the structures in place. So yeah, that's, some people can.
So, Maria, I know that it's not, it's very rarely, but women without even fallopian tubes, they would, maybe they were removed or they were tied, they still can get pregnant.
It's very rare.
Yes.
And another really, really rare thing that can happen is fertilization can take place outside the fallopian tubes. Because think about the travel. Okay? Here's a deposit of sperm. Sperm swims up the tubes and goes out the tubes. Well, if they're still viable at the end, they're going to keep swimming. Yes? Chances are they're not, but it can happen. And what if this guy misses? There could be fertilization that takes place out here. So again, very, very rare, but I bet you if you go back and look at medical cases, it's happened. No, then you're in big trouble because that cell, although it might stop, it might stop its replication process and die. But yeah, that can happen too. Now, as far as no fallopian tubes, again, big shot in the dark, but if for some reason when ovulation, say it takes place over here in the tube, there's a small short section over here that happens to be clear.
How clear?
I don't know. But basically what you have to be able to do is fertilize that oocyte and then have it tumble down and implant in the uterus. So I can't answer that question.
Generally speaking, because I have heard of cases where women who have undergone, having their tubes tied, have conceived.
Yep.
That sometimes is a medical mistake, right? Because
It could be.
They may not have been
Cauterized properly.
Right.
Because typically when they do the procedure, they cut and cauterize. Also, same thing with males and vasectomies. They're magical babies. But sometimes that procedure wasn't effective. So, or that the tube regrows and connects and is open.
Don't they sometimes, isn't it possible to have it reversed?
Yeah.
Yes. And basically they're going to take the tubes and put them back together.
So, which structure inside the fallopian tube will help those not cell, oocyte and
Peristalsis.
peristalsis?
Yep.
But somebody told me that there is a cilia or
Well, it's similar to what we saw when we discussed moving through the small intestine.
yeah.
So same.
So there's some cilia that helping them smooth out from the tubes to.
I don't know the answer to that question. Nope, I don't know. But I know peristalsis takes place. I don't know if the cells are ciliated. So once fertilization takes place, we want to maintain the endometrium, and we want to get that cell down to the endometrial layer of the uterus. So certain hormones are going to help maintain after implantation that endometrial layer so we don't shed it. One of those hormones, very important, is something called human chorionic gonadotropin hormone. So you got, human chorionic gonadotropin. It's on page 1070, bottom paragraph in bold black letters. And you should already know this because you talked about it in lab, I hope. Okay, and what was that hormone that we test at certain levels to see whether implantation has taken place? HCG. So once HCG levels reach a certain point, you'll start spilling them over into your urine, and you can actually test to see whether you're pregnant. That's what you do when you pee on the stick. So we're actually testing for this hormone. You can do blood tests too. Well, no, it's actually a chorionic hormone. Chorion is before the placenta, and this is where we're establishing a lot of the blood vessels. So it's a chorionic hormone. So I do want you to understand or know these terms from this diagram. So day 1, fertilization. After day 2, we have cellular split. So we have a ball of cells, usually, again, different for different people. We're at the 4-cell cycle. By the time we hit 3 days, only 1 day later, mitosis has gone crazy, and we're in the solid ball phase. Yep, we call it a blastomere or a morula at the 3-day phase. By the time we start entering into the uterine region, it's about 4 days. And what's going to start happening to that ball of solid cells is it's going to start to differentiate. And this is where we start to form different layers of cells, which will eventually become different parts of you as an adult. Like some of those cells will become your nervous system, and some of those cells will become your digestive system. So we start to differentiate this early in the cellular division phase. So on day 4, when we're just starting to enter the uterine area, we call you a blastocyst. And this is where we start to see differentiation of cellular material. We get rid of that zona pellucida at this point, and we start to see differentiation. By the time day 7 takes place, and day 7, it takes 1 whole week for that ball of cells to fall from where it was fertilized to where it's going to implant. On day 7, we call you a blastocyst. And this is where we start to see very distinct layers of cells. The trophoblast on the outside, we start to see a space or a cavity called the Blastocyst Cavity, and then we start to see a group, inner group, or inner cell mass. And again, this is going to then differentiate into different cellular layers as those cells continue to reproduce. Now that implantation has taken place, it's important for us to establish a feed from mother to developing zygote. So some of those cells, and those outer cells are the culprits, are going to start to form a group of feeding tissue called the chorion.
How it's called?
Chorion. Another thing that we're going to see is changes in hormones during gestation. I know that they kind of throw this in here a little bit early, but since it's here in this order, I'll throw it at you now. So ovulation, fertilization, that's sort of day 1 here with respect to pregnancy. We're going to start to see a rise in human chorionic gonadotropin. And that's going to continue rising until about just after what? Yeah, and this is weeks of gestation, okay? Not days, weeks. So we've already had fertilization take place. We're already starting to establish that connection, which we're going to talk about in a moment. And we'll see the peak at around 8 weeks of pregnancy of human chorionic gonadotropin hormone. So sometimes when I suspect I'm pregnant, I can also go to the doctors, right? And I can have a blood test. What are they doing? Yeah, this is a more specific way of seeing where you are by testing the exact levels of HCG in blood. So that's a more exact way of telling how pregnant you are.
If you have miscarriage, you can tell by that too.
Exactly. So start to see that drop way too soon. Okay? So HCG, near the end, we start to see on board a lot more who? Estrogen and progesterone. So they're going to be, we're going to look at them closer to the last weeks of gestation. How many weeks gestation? 40 weeks. 40 weeks. Not 9 months. 40 weeks. And if you tell a woman she's going to be pregnant for 9 months, you better run, because it's 40 weeks. And when she's in her 38th week, yeah, watch out. Why? Look at the hormones.
Someone I know is delivering her baby today. And
Oh, wow.
42 weeks.
Oh, boy. Well, and again, it's not exact. Like we use these levels, but they're not always exactly the same in all women. So it's very difficult to predict that exact gestation.
According to the
Bec-
doctor, I had my daughter at 36 weeks, and I was at least 38, because we knew the exact date of conception, like that it happened, and they were always like
Yeah.
months off.
And again, it's not an exact science. And we metabolize things in different ways and secrete things in different ways. So, yeah.
But I used to laugh at her when she was like, "You're gonna have." My due date was at the end of November, and I had my daughter at the end of October. I was like, "I'm not gonna make it to November."
We know better. Okay, so day 7.5, day 8, we see implantation happening. Now we have to establish blood supply to these developing cells. In the meantime, what are we going to have there that's going to supply nutrients to these cells as they divide? Because we still have a ball of cells that needs nutrients. Well, the endometrial layer is going to provide some things, but while we're establishing the chorion, we actually have something called a yolk sac. Who has yolks? Eggs. Eggs like in chickens, and eggs like in lizards, and eggs like in snakes. I don't know. Just like an animal. This is where we see, and there's a big science called embryology, where we study, as an embryo is growing, the similarities in the embryo between a human and so many other organisms on this planet. So evolutionary biologists and embryologists study those similarities. But that yolk sac will eventually become a place, no, not quite the placenta, but it'll eventually become a place where we're going to make our first blood cells. So later on, as we see later on development, that yolk sac is going to become part of how we produce blood cells for the new developing fetus. The chorion and chorionic villi will eventually establish themselves into what? That's going to be your placenta. So that group of cells, that network, is going to basically, through chemicals, draw in and cause the production of the capillary network, which will eventually become the placenta. Excuse me.
So yolk sac and which?
Yolk sac will eventually become a group of cells that will eventually be your blood cells. But before that time, so when we're talking about day 7 to day 16 to 4.5, actually to day 16, we're going to see nutrients coming from that yolk sac for those developing cells. We're going to continue to start to differentiate. And now in the diagram, you start to see different colors. Does everybody see those? Those colors are going to delineate the different cell layers. Yeah, good. It's over here. If you look forward in your textbook on page 1076, figure 28.10, you see those same colors coming into play. Because these are the layers that are going to be formed of different cells, which will then differentiate into some of your different tissues and organ systems. So we'll revisit those colors again. So by day 16, we've started to establish all of the beginnings of blood vessels that will eventually become the placenta. The chorionic villi are always going to be there as well, and we can actually do testing on that chorionic villi to look at the genetics of the developing embryo as well.
So what's the chorionic villi?
It's basically the beginnings of blood vessels that will be established or establish the placenta. So if you look in the diagram at week 13, what happened to the chorionic villi? Yeah, it starts to shrink down and become part of the placenta that's formed. So the placenta is what? Well, it's basically it's a circulatory connection between the developing fetus and embryo and fetus, and mother. So you start working before that child is even born, because you have to do what?
Provide nutrition.
Yeah, provide nutrition. So your circulatory system, besides providing nutrients to your cells, are also through the placenta that's been established, providing nutrients to the developing, what do we call it now? Fetus. So at week 13, again in this textbook, you'll see it different in different textbooks. At week 13, we now call you the fetus, because what has been established and matured. That placenta has been established and matured. So we not only provide nutrients to the developing fetus, but we also take care of what? Well, ourselves too, hopefully. But we also take care of getting rid of what for the developing fetus? Waste products.
Maria, what there's a lot of definitions. Do we need to know all from that one? Because there's a lot of like.
Yeah, I just basically want you to know the different time frames. So implantation, 7.5, 7 days, we have a blastocyst. By 12 days, we start to form and establish that chorion.
The time frame in this book.
Correct. Again, you're going to see it different in different places. And it is different, I'll warn you. Huh?
A fetus is 8,
It depends.
but it's 13.
No, 13 weeks in this book. So learn this. It is, it's different. I'm warning you. Yeah.
But it says in the beginning of the book it says 8.
Well, 3 months, 2 months in some
It
places.
says in the first
It says
page.
9 weeks to birth is the fetus.
Yeah.
From fertilization through week 8, embryonic period is called embryo, and from week 9 through birth, the fetal period.
Yeah.
and conception
So 2
is
months.
fetus. It's on page 1065.
Yep. So 2 months, right?
Okay.
So when we're in fetal stage, we've established the, thank you. And that's what I need you to know from that diagram.
So no other details.
Nope. Yolk sac, you should understand what the yolk sac is. You should know what a blastocyst is and all the other stuff that we've already mentioned from the other diagrams. You should understand what the chorion is. You should understand what the placenta is.
For my girls, I have been taking some of this, so I guess I looked on the internet and it seems like it attaches itself to the uterus and sometimes can grow out.
Yeah, or cover the cervix. It can do all sorts of weird things.
It stayed inside the disease for them.
Yeah.
I almost died for a second.
Yeah.
Hemorrhage.
So when she has to pass it, you have to pass the placenta.
Freaking party, the whole thing.
Yeah.
They're like, get that out, and I'm like, ew.
I thought it was done. Okay, so the next part of the book talks about the events of embryonic development, gastrula to fetus. So, no, not that. So if we look at this diagram, we see the formation of the layers that are starting to come into play. And these are called germ layers. And there's 3 different layers called primary germ layers. First, we see fluid starting to form in some of the spaces that were created in the beginning. And we start to see differentiation of cells. So there's 3 different layers, which will eventually become 3 different systems in our bodies. We see the ectoderm, and the best place to look is down here to see all 3 layers. We see a mesoderm, and we see an endoderm. So as we go on through day 16, we continue with differentiation and the formation of these different layers. A better diagram that I kind of like is the one on page 1076, where we see the differentiation of those layers and what they will truly become. So understand what the 3 layers are: ectoderm, mesoderm, and endoderm. And in your textbook, starting on page 1077, we see a description of what they will become. So specialization of the ectoderm. What is the ectoderm going to become? Yeah, your nervous system and your skin. How about mesoderm?
Where do you see that?
On page 1077.
And when it says which one will become which?
You're correct.
No, I don't see that. That's why I'm asking.
It says it right under each one of them. Specialization of endoderm,
Okay.
specialization of ectoderm,
I see. I thought
and specialization of mesoderm. Okay? So basically in this picture we're showing you what happens in the 5-week embryo and where some of these different parts are. Okay?
we need to know where, which
You need to know what each layer will become. I think there's a chart. Oh yeah. On page 1079, it's an easier visual. It shows you what the 3 layers will become. So in this diagram, we see that at 5 weeks, an embryo is starting to have the beginnings of what we see in the adult. So we see a pharynx. We see the beginnings of the thyroid and parathyroid and the thymus. We see the trachea. We see the beginnings of lungs. We see the beginnings of stomach, the liver, the pancreas. That's 5 weeks. When we look at this diagram, again, showing you exactly what those cells are becoming and when they're starting to differentiate and move to the places where they will eventually arise into these different organ systems. So at day 17 versus day 20 versus day 22, by the end of 4 weeks, we see our little friend in the diagram previous. So this diagram, again, I'm not going to make you know specific details, general details of the 3 germ layers. So ectoderm, this is a nice diagram because it gives you a nice flow chart of what everything will become. The mesoderm and the endoderm. So this is a nicer one. I like this diagram better because it spells it out for us. When you discussed the circulatory system, or we discussed the circulatory system, one of the things that I hope was pointed out in lab was the difference between the circulatory system of a fetus versus the circulatory system of a newborn and eventually adult. Now think about the development of these organs. In order to develop the organs, in order for the cells to reproduce, they need nutrients from where we're getting them from. When we're a fetus, where are we getting our goodies from? We're getting them from mom. The other thing we get from mom is our respiratory gases as well. So we have lungs, and basically we have to get the lungs nutrients, but are the lungs doing anything for us at this point? No, because where are we getting our respiratory gases from? Not breathing, because we're hanging out in an amniotic pool. We're getting them from maternal circulation. But again, we still have to get blood to the lungs for development. So when we look at this diagram, we see the circulatory system of a fetus versus the circulatory system of a newborn. What are the differences, huh? Right. So we have something called an umbilical artery, and we also have something called an umbilical vein, right? When you dissected your pig, or you're in the process, right? Did you find the umbilical arteries? Okay. Pay close attention. When you look at the umbilical cord of your fetal pig, it's going to be kind of weird because in the fetal pig we see this central pouch, which is the urinary bladder. Because what else does mother have to do for baby? You got to get rid of waste. So there's a connection there in the umbilical cord of a fetal pig. Remember, that pig has never been born. So mom's still taking care of business. The two tubes that run beside the bladder are umbilical arteries. So pay attention to, did he give you that little handout, little blue thing? Okay. Okay. That's a nice diagram. It should be up on your website too. It's kind of blown up and sometimes people have a hard time printing it off. But notice the makeup there. And that's going to be in the umbilical cord. So we have umbilical artery, umbilical vein. Again, mother's connection doing all the work for baby already. And we also have to get rid of waste products, so there's a connection there as well. Those connections, those tubes, are internal in the child as well. So outside the placenta we have our connection, and those tubes continue on inside internally as the fetus develops. The other difference we're going to see is, remember circulatory system? We came into the atria, we left out the ventricles, and the heart was sort of split in half. The right side of the heart was called the what?
What do you mean?
The right side of the heart was called the pulmonary pump, right? Because where do we have to? We got blood in from the whole body, did its thing, and then we had to pump it to the lungs, right? To get what? Exchange oxygen, carbon dioxide. We still have to do that, but not for the same reasons, because the lungs isn't going to do that for me in the fetus. We still have to get blood to the lungs to feed it. Exactly, but we don't need to put the whole volume there for gas exchange. So what we're going to see is a hole between what? In the fetus. Yeah, the two atria. Yes, sometimes it doesn't. That's called the, what is it called during fetal development? It's called a hole, a fossa ovalis, or what's it called? Foramen. It's a foramen, right? And yes, I'm telling you right now. So the Foramen Ovale is between, no, and basically it's just the things in blue on this diagram. And you already know it. So we see a Foramen Ovale, which will then become, and see the little arrow? When the baby's born, it's going to become what? It's going to become the fossa ovalis. So foramen, when the baby is developing, fetus, it's a hole. That hole has to do what? It's got to close up so we can get the normal circulation going in the newborn. So in this diagram, you need to know the stuff in blue, yes? And understand that the top is in the fetus and the bottom is in the newborn. So a lot of these extra tubes and holes and things like that, some of the tubes will basically become ligaments. They shrink up and they become a ligament that sort of holds things in place. The big one is the one that's feeding into the liver, and that's the umbilical vein. When you dissect your fetal pig, you're going to see that connection. Before you cut it, when you open up into the body cavity, you're going to see those tubes, the umbilical veins, that will run into the liver. You're going to have to cut them so you can pull them down and look at those abdominal pelvic organs. But that umbilical vein is going to become something called the ligamentum teres. And you can see it there in the blue box. And that's basically a ligament that's going to help hold the liver in place in the newborn baby and you as an adult. The umbilical arteries are then going to become ligaments as well. And they're basically just holding parts in place. They no longer have function as a circulatory organ, but they're ligaments that help to hold things in place in the cavities. So basically know from this diagram the difference between what those parts are in the fetus versus what they are in the newborn. Are we good? So there's a whole bunch of really, really neat films on fetal development that you can watch. One, it's kind of old. It's a Nova film, but it's called, I think it's called The Miracle of Life. There's some really neat footage on fetal development. But the events of fetal development show us a whole bunch of different changes in a very short time throughout the pregnancy. So we will pick up with events of fetal development on Tuesday. So here's what I suspect will happen. Are you ready for this? This is the rest of the semester. We're going to finish this fairly quickly on Tuesday of next week.
But I don't think we will be able to.
Mila, we're going to start what? On Tuesday of next week. We're going to start heredity. We'll hopefully get really close to finishing that, but probably not. We'll finish up heredity on Thursday of next week. Okay? We will have, and maybe I'll start a little quiz session on that class Thursday. Just throw out some PowerPoint questions, not PowerPoint, but clicker questions, and review if we have time left in that class. On Tuesday, the final week of classes, Tuesday we'll have our exam on those 5 chapters. And then the last day of classes, that Thursday, that will be your cumulative final exam. I hope to be able to get through that PowerPoint extravaganza that I've created, but if I don't, you know what I'll do? I'll put it up for you. Okay?
What is it? Is it quizzes or?
It's just a whole bunch of PowerPoint questions that, yeah, it's just a review. Remember, your final exam isn't something you're going to sit down and cram for, because you can't.
You already know.
You should know it, exactly. So at that point, and remember, I only have an hour and a half to give you an exam. It's not going to be as detailed as the tests are. It's going to be very general information that you should know. Okay? So don't panic about the final. What should you do to study for the final? Nothing. Exactly. Relax. Meditate. Breathe. Don't try to cram for that final. It's just useless.
Are we going to have a quiz due Tuesday?
Correct.